Hi everyone. My name is Lee Joon. You can also call me LJ. I'm a postdoc working at the Civil Beamline. And as Greg mentioned, this talk is about lipid nanoparticles. And specifically, I'm going to talk about how we've been using our HT-SAXS platform to characterize lipid nanoparticles for vaccine development and design. So to talk about our motivation behind studying lipid nanoparticles, I'm going to bring up COVID-19 pandemic, which was a huge global threat to human health. As of February 2024, over 774 million confirmed cases have occurred, and that has resulted in over 7 million deaths. And I always like showing this weekly COVID-19 cases and deaths because it's very easy to highlight some important dates here. This only runs up to, I think, October of 2022. So it's missing a lot of the current data, but we have the first release of the vaccine in 2020, at the end of 2020, and then FDA approval in August 2021. And as the vaccine started rolling out, even if the number of cases increased, the number of deaths coming from these cases remained relatively lower compared to earlier on. And to just highlight our huge success during COVID-19 in terms of the field of vaccine development, here are two graphs that show like percent total population with at least one dose of the vaccines in the US and of the world. And actually in numbers as of February 2024, over 13.5 billion vaccine doses have been administered, which is a pretty impressive number. So how do the vaccines work? We have the coronavirus with the spike proteins on the surface. And so we put inside these lipid nanoparticles, the mRNA molecules which code for the spike proteins. And then the lipid nanoparticles, I'll also be referring to them as LNPs throughout my talk, are what's responsible for safely and effectively delivering the actual active drug components, which are the mRNA molecules inside the human cells, where they can be translated into viral spike proteins and then trigger our immune response. So when we think about designing good vaccines, we want to think about designing good lipid nanoparticles, but there are a lot of challenges that come with that. So here's a very brief, very simplified overview for those of you who are not familiar with how the process of formulation occurs for LNPs. We have the lipid solution in ethanol and oligonucleotide solution in acidic buffer, and then we mix these two solutions together to generate these lipid nanoparticles that can be assessed for particle size, distribution, encapsulation efficiency, and more. And in each of these different steps, there are a number of factors that affect the structure and also the efficacy. So I've highlighted just a few here, and these include things like lipid compositions, type of nucleic acid, mixing method, and also some environmental conditions like presence of additives or storage conditions. So there's a lot of variables that come with each step. Just to highlight a couple, and you'll hear more about this later on in my talk, there's a variability that comes from formulation method. So depending on which method you use to mix the two solutions together, you can end up with different results. You might get larger and more variably sized LNPs. You might also get to smaller and more uniformly sized LNPs depending on which one you use. And you can also get structural variability from chemical compositions. And so these include things like what's contained inside the lipid solution in ethanol. This is really a cocktail of various lipids. We have cationic ionizable lipids, structural helper lipids, cholesterol, and PEG lipids. And all of these lipids are essential because they play a different role as in terms of like what they do inside our body, how it's structured. So depending on which types of lipids you use for each of these various classes, and the relative molar ratios of these lipids to one another, you can end up with a variety of different structure and efficacy results. So I've just pulled a couple examples from literature to showcase that structural diversity, even though at first glance it looks like they're all just circular molecules or particles. We can have particles that have a monolayer around the particles, or you can also have what we call multilamellar that look like stacks of onion rings. That's on the top right. And we can have these core regions that are more disordered. So these lipids and nucleic acids are sort of just arranged in a more random manner. You can also have the core that's a more in a more ordered structure at the bottom right-hand corner that we call an inverse hexagonal core. And we have these nucleic acids that are arranged in a more structured manner. And previous studies have suggested or concluded that internal structure affects in vivo delivery and uptake, where if you have a more ordered core structure, that leads to better activity. So it becomes essential for us to really start characterizing the internal structure of these lipid nanoparticles and start linking them to efficacy and their formulation in order for us to better design vaccines. So again, as I've just mentioned, we have all three of these factors that really we want to take into consideration. So that's formulation, structural analysis, and efficacy. But because of the high variability that comes from all of these various factors involved in formulating these particles, it's pretty hard for us to go about in a rational way to start designing these lipid nanoparticles. So a possible solution here is to use a high throughput screening workflow to try to hit as many of these variables and factors as much as possible, and just building up that database of what creates better lipid nanoparticles, what creates better vaccines. So in pharmaceutical industry, so that's one of my, I guess, my major collaborators. As I'm working at RB9, they have a high throughput screening workflow for LNPs, where they take the two solutions that I mentioned before and then mix them together to generate libraries of lipid nanoparticles, and then they can assess that for drug delivery efficacy. And this here, as opposed to the vaccines that I mentioned earlier, are loaded with antisense oligonucleotides, so just keep that in mind. And they have shown that this can be scaled up with translatable results. But as I mentioned earlier, in this gear, all three gears working together, they're lacking that structural analysis component in this high throughput screening workflow. So that's where we come in as a beamline and me as a collaborator with industry to trying to supplement their current working pipeline with that essential internal structure data. So Greg did a great job of going through the background for small X-ray scattering, so I'm not going to go through this in much detail, but we have the diagram of like our setup at the top and then the physical setup at our beamline at the bottom. I get my samples from my collaborators in the form of 96 wall plates, since I utilize the high throughput aspect of our beamline. And then we have our sample cell in the form of a needle that Greg showed briefly earlier that will pick up our samples from the 96 wall plate to the path of the beam where we can start collecting data. So I also have a big video clip that play without sound, that shows sort of in real time what that looks like. So we have the needle reaching into one of the wells of our 96 well plate, it's picking up our sample and then moving it over to the path of the beam, and it just kind of repeats itself. And we are able to run, I think, a full 96 well plate in a little over an hour. So this works quite well for me and for my collaborators in terms of testing various samples in a short amount of time with, I want to say, pretty, like, we can do this in a pretty automated way. I'm pretty happy with the workflow for data collection. And so once that beam gets scattered, it will hit the detector on the right, which I've also highlighted down here. we have an example of a detector image and the resulting scattering profile that I typically see with these samples. And from these, we are able to access various information like internal structure, shape, and size. And because of our high throughput aspect, we're able to test a lot of different experimental parameters, which is important for us to start hitting like what factors affect LMPs in different ways. So we're able to observe trends while varying all of these experimental parameters utilizing our high throughput platform. And so an example of that I'm going to go through is this recent application and comparing lipid nanoparticle formulation methods. And here is the citation for that if you're interested in checking it out. Here I mentioned before that we can get some structural variability coming from formulation methods. So we're comparing two different methods, one being the coaxial turbulent jet mixer at the top, and then the microfluidics method at the bottom. And these two methods all have their own different pros and cons. I would say the main advantage for the turbulent jet mixer is the material, the stainless steel material for broad solvent compatibility. And the main limitation being you require a larger amount of material for each experiment. And then for microfluidics, it's pretty well established now, so there's a good commercial availability, and you don't require as much material per experiment, and you can do this with very minimal human intervention. That being said, though, you do require single-use disposable cartridges for microfluidics, so then that tends to add up in terms of cost and also waste. And then you do get some variability going from cartridge to cartridge. So that's something that we want to look at as well. For the actual design of experiments for our high throughput SAXS platform or screening, we had 54 different lipid nanoparticles that were all loaded with antisense oligonucleotides with this lipid composition. The main factors that we were varying were flow rate ratio and the ratio between lipids to oligonucleotides that I call N over P, or we call N over P ratio, and the content of our ionizable lipid, MC3. And the flow rate ratio, we were varying because if we have faster mixing speed, that tends to lead to smaller lipid nanoparticles, and that also has been linked to better encapsulation efficiency. And so we were interested in seeing how that affects the internal structure and of these particles. So I did like a very simple analysis for this paper where I was mainly just interested in looking at peak center coming from the scattering profiles. So I identified two different regions, one being the disorder region at the left, highlighted in blue, and then the order region being in orange, a little more to the right. And you can see that with the order region, the peaks tend to be a little sharper, less broad compared to the disorder region on the left. And we were able to start comparing the jet mixer method versus the microfluidic mixer, looking at how these shift from one another. And we did this for all 54 lipid nanoparticles and complemented that with various other techniques to try to observe these key trends or key metrics. We supplemented our SAXS data with dynamic light scattering, or DLS, for looking at particle size, and then encapsulation efficiency, looking at how much of the nucleic acid was contained inside these lipid nanoparticles, and then we also used cryo-EM. And I'm not going to go through the actual figures here in too much detail. If you are interested, there is the paper cited at the bottom, or feel free to grab me after and ask me about it. But general trends that we observed were if we increase the amount of lipid to nucleic acid ratio, we saw a peak shift going from higher to lower Q, which indicated a loss of internal order. And we presume that this was due to increased lipid to lipid interaction that led to more disordered core inside these particles as opposed to lipid to nucleic acid interaction that leads to these encapsulation little pockets. And then we were able to utilize correlation plots looking at various metrics of our two different types of sample mixing methods. So from the coaxial turbulent jet mixer, we saw generally there are smaller particle sizes, narrower particle size distribution, better azo encapsulation. Microfluidics retained better internal structure order, and then we saw fewer aqueous blebs that we observed under cryo-EM conditions. So then that led us to conclude that turbulent jet mixer leads to faster mixing, and this could potentially save cost on nucleic acids if we were able to reduce the volume requirements for these experiments. But as I mentioned earlier, we do want to take into account formulation and structure and efficacy together, and we haven't really done the efficacy part of that here, so we will still need to go back and run more experiments to compare functionality. But this was a good example of how we can utilize the high throughput aspect of our beamline to try to look at the one of the important factors of formulating lipid nanoparticles, which was the actual mixing method. So that was an example, but we were also interested in potentially extracting even more information from these scattering profiles as opposed to just looking at the peak centers and peak shifts. So that's where the next example comes in, and this was actually worked on by Mikhail, who's sitting right here, where we were able to apply high throughput SAXS to lipid nanoparticle workflow and start assigning more of those key features to the structures of these LNPs. So we have a graph here of a LNP sample going from lower to higher concentration. And as we increase concentration, we can see these features that stand out more. So for example, this one, oh my gosh, this main peak looks like a single peak at first, but as we increase the concentration of the sample, we see that this is actually containing two peaks shown from the shoulder on the right. And from the ancillary peaks on the right, we can assign this left peak in pink to be inverse hexagonal and the right one to be lamellar. And we are able to also double check that the distances actually match up with what we expect coming from, for example, lipid bilayer, 45 angstrom. And then we can also see that the region on the left corresponding to the disorder region also increases as we increase concentration. So this is that broad signal that I was alluding to earlier in that example. And we are able to verify that these assignments match up with what we see under cryo-EM. So I've highlighted the regions that correspond to each of these structural features are highlighted at the top right, and then are in this schematic at the bottom right, where we have that lamellar feature coming from the association of the nucleic acids sort of sandwiched inside the lipid bilayers. And then we have the inverse hexagonal base coming from these nucleic acids arranged in a little ordered manner inside these compartments. And then the disordered core region where that ordered organization kind of breaks up. Again, utilizing our high throughput platform, we are able to start screening different variables, and in this case we were looking at the types of PEG lipids and the amount of PEG lipids relative to other lipid molecules. So this is again 54 different LNPs, and we chose a couple of these particles for cryo-EM just to make sure that we were able to confirm what we saw under the scattering profiles. And then going back to what I was mentioning earlier about potentially extracting even more information from these types of data, we were able to deconvolute the SAXS scattering profile. So this is a region that we're particularly interested in, a Q of 0.1 to 0.215-ish, where we use 3 Lorentz functions to fit the data, and each of the Lorentz functions are responsible for or representative of each of the different phases that I mentioned earlier. So that's disordered, inverse hexagonal, and lamellar. And those are highlighted in the 3 colors underneath the main scattering profile. And once we are able to do that, we can start quantifying the amount of azo to lipid compartmentalization and then use that for further analysis and trying to look at how efficacy might be correlated with the internal structure, which is this correlation plot on the right. We have the x-axis coming from our SAXS data of order and disorder, and then the y-axis from our collaborators who ran the efficacy, the bioassays. And then we can see that between the lowest 15 and the highest 15% of the SAXS ordered metric, we see a fourfold difference of the efficacy. So this was a pretty significant finding for us, and led us to conclude that internal structure is correlated with azo efficacy, and ordered particle core does favor enhanced mRNA knockdown. Another way we can visualize that is using a heat map. So this is the data just visualized in this format, and we can start pulling some interesting findings. So left panel here is from our collaborators, the efficacy studies, and then the right side from our HT-SAXS data. And using this, we can start pulling more specific information about how these different PEG lipids have an impact on the internal structure and efficacy of these particles. So that was an example of going through from formulation to structure and efficacy, where we are really starting to pull out more and more information from our HT-SAXS data to complement their existing pipeline in the pharmaceutical industry. And then I have a really quick example. That's the most recent application of using our platform to study these molecules, where we were looking at storage conditions of the mRNA LNPs. So the mRNA LNPs do need help preserving their integrity, and because of various reasons, like mRNA being prone to degradation in solution or in higher temperatures. And then the lipid nanoparticles themselves are comprised of complex intermolecular interactions and can be very sensitive to environmental changes. So then the ultra cold storage does, it really serves the purpose of preventing aggregation and maintaining the encapsulation level of the mRNA within the lipid nanoparticles. But that being said, it does pose a challenge for distribution for countries, especially if they don't have sufficient infrastructures. As you can see on the map on the bottom right, the color scheme of the percent of total population that's received at least one dose of vaccines is not the most even as you might like. So then lyophilization becomes an alternative strategy, and so that is freezing the sample and then just stripping away moisture. And previous studies have shown that this leads to pretty stable lipid nanoparticles that can last for 6 months, either at 4 degrees or 25 degrees. And I am running out of time, so I'm gonna go through this a little more quickly. And each of these steps do also pose a lot of stress for these particles. So then it's important for us to look at lyoprotectants and buffer conditions to preserve as much of the integrity as much as possible. So then that's what we were doing with this most recent example. We have 45 lipid nanoparticles loaded with mRNA, and we were using the Moderna formulation here and screening a bunch of buffer lyoprotectant types and concentrations to see if we can identify some optimal conditions for lyophilization. And just for fun, I have an example of what these cakes look like afterwards, which look like little discs with different colors. Pretty similarly to my first example, we complemented this with a lot of different techniques to trying to correlate with activity and structure. Generally with increased particle size, that leads to poorer encapsulation efficiency, less intact mRNA. But I want to dive more into the structure of how we were able to utilize SAXS and cryo-TM together. And we saw good agreements going from SAXS and cryo-TM, where if we observed internal structure being retained under SAXS, we saw that also in the micrographs. And then we can start tracking how it changes going from fresh sample compared to the lyophilized sample. And this is again the same 3 Lorentz function fitting approach that I've shown earlier. So here, general trends that we observed were a good match between the two structural techniques. Most of the lyophilized mRNA LNPs had more disorder inside, which makes sense as it sort of reorganizes itself, it loses order. Interestingly, we did see some buffer effects where acetate tended to be better than the other two. And it was very difficult for us to observe a straightforward correlation between activity and structure compared to our second example. So further studies are needed to try to tease that out from this screening. So here we were able to explore a pretty wide range of conditions like lyoprotectants, concentrations, and buffer. And that really highlights the power of our platform, where we are able to look at all of those different factors and variables in a single set of experiments. And we were able to observe strong correlation between SAXS and cryo-TM data. So that also was a positive result for us. And as we continue to generate more and more data from our high throughput platform, we saw this need to create a unified database for LNPs. So we have a website, we have an open data repository, sample scattering, where we are starting to deposit more and more of these lipid nanoparticle data sets, and in the hopes that in the future, we can come in with AI-assisted algorithm, for example, to try to better design lipid nanoparticles and also vaccines. So that's really all I have for today. I had 3 main examples of how we utilize our SAXS platform to characterize lipid nanoparticles so that we can start identifying these effects of these various factors that's involved in formulation conditions and how that affects structure and efficacy. And then I gave a little plug at the end about our data repository. So with that, thank you all for your attention, and I would like to also acknowledge everybody that's been involved. There's been a lot of collaborators. I'll take any questions.
Questions? Check on your chat. Nothing in chat.
Despite the fact that you mentioned the lyophilized samples are less efficient, are still a good option?
I'm sorry, what?
Are still a good option, the lyophilized samples?
Yeah.
For to be used, right?
Yeah, yeah.
What is the difference between this and the not lyophilized, the efficiency?
Oh, so that was very interesting because they. So I think this one is probably best spot for that. So I didn't get to explain this in detail, but we have the change of the order, internal structure, and then the change of in vitro activity. And we have the fresh sample right here. That's pre-lyophilization. And that's sort of like our base, like the center point. And so we see that there are a lot of different changes in the activity after lyophilization, and it's not super clear of like why conditions, like how the conditions are really correlated with the change in activity and also the change of structure. So that's definitely something that we want to look at more.
Okay. Yeah, but in structure-wise, that peak disappears.
Yeah, that was very interesting. Yeah. So yeah, a lot of I think in a lot of the cases, even if they lose internal structure and sometimes very little structure that I can still see getting retained after lyophilization, in some cases they saw like absolutely no change of activity. So it was still really effective, even though internally the structure wasn't there anymore or as visible.
So that top left 20% sucrose tris, that's fresh, and then right below it is after lyophilization on the left. No, I'm talking about the SAXS data and the cryo-EM data.
Oh, this one, yeah.
So those are the 2 comparisons. That's typical, like change. So that's protected by 20% sucrose in tris, And then you can see the cryo-EM, it kind of like expands, like the tubes, yeah, they get big. And some of after lyophilization, there's actually some cases where the apparent efficacy increases.
Yeah.
But in general, the perspectives are good with that.
In general, the activity-wise, it's good, and also the physical chemical properties, they correlate very well with efficacy. Yeah, it's just that with the structure and the efficacy, there's definitely something more going on. Yeah, so should be exciting. Yeah.
And these weren't live like mouse studies; these were just like there was a cell-based study. But again, when you inject this stuff into your body, right? Yeah, everything changes, right? So we can characterize up to a certain point in terms of efficacy.
Yeah, no, thank you for the question. Hello.
When these LNPs are being manufactured at an industrial scale by Moderna, are they using a jet method or a microfluidics method or some other method or do you know?
I think the most common is still microfluidics.
And okay.
Yeah.
So you can produce the quantities needed.
Yeah. In fact, when they were testing that scalability, that was done using microfluidics. And I think they just scaled it up with like their setup. And the jets, the coaxial turbulent jet mixer that I introduced earlier, with comparing between microfluidics and the jet mixer, that actually has been used a lot. I think used for liposomes, but not as much for lipid nanoparticles. So that's why it was like more of a newer method for that. And they wanted to see if it produced LNPs as well as the very well established microfluidics.
Microfluidics is the one that's been around and is in more common use.
Yeah, and you can also just like pipette like the mixture up and down too. It just leads to like very not uniform size. Yeah. Any other questions? Oh, I can't see those names from here.
Nothing in chat that I could observe. And yeah, I mean, that strong peak obviously is from the organization of the lipid layers, right? So
yeah.
Nice different organizations within the LPs.
Yeah, it kind of helped that like I saw the shoulders again, like in this case, for example. So it helped to identify that there was a second peak
phase
under. Yeah. Yeah. And then also, I do notice some distance changes going from, because previously it was the antisense oligonucleotides and these are mRNA. So then that distance of the peak, where the peak centers are located, it does also shift, yeah.
And relative to protein SAXS, right, these are going to be heterogeneous, right? You're going to have different sizes, you're going to have different things, so not pure one thing. So it's a little more complicated. Each of them isn't identical necessarily.
Yeah. So we have it easier.
Yes. Yes.
The lamella should be consistent.
The lamella should be pretty consistent. Although if you use different, like it does also matter because like what if the lipid bilayer like that size is different, so then that will also change. So yeah, I always, it does help to have like cryo-EM next to it, but it is a lot lower throughput. So I mainly pick out the very representative particles, like things that just look very odd under scattering profiles, and then they'll run the cryo-EM for those, and then I usually can verify, okay, so what I was seeing in SAXS, what I was thinking, what was happening is actually what's happening. It's just like a confirmation thing. Yeah. So it helps a lot in terms of throughput.
Right.
Oh, sorry, yeah.
Yeah, how much, like with the microfluidics, like how much change if you alter the velocity through the channels, or
Wait, so your question is how much change of
Yeah, like for your nanolipids come out after, say you introduce it at just high rate, high pressure.
I guess how does flow rate affect structure?
Oh, okay. So yeah, so I think generally if you increase the flow rate, it leads the mixing occurs like more quickly, and then that tends to also start that encapsulation process more quickly. So then I want to say at least a smaller size, unless you like leave it out and then it starts like reorganizing itself, and then so things might like combine together and that creates like a larger particle. But generally with the flow rate ratio that we were looking at, it's if you increase the flow rate, then that leads to smaller LNPs with more encapsulation of the lipid, of the nucleic acids in the lipid compartments. Does that answer the question?
Yeah.
Perfect, thank you.
Right.
Any other questions? Okay. Cool, thank you.
Thank you for your time.